So let's what we were doing. What we finished was we finished chapter 7, which was DNA structure and its replication, and we start out with DNA structure, understanding it, understanding that A's bind to T's, G's bind to C's. Talked about nucleotides, backbone, hydrogen bonding, and then we move into its replication in humans and how DNA is replicated, as well as how it's replicated in a laboratory, for example, using PCR, which is the polymerase chain reaction. We talked about how then that breaks, how it goes through a series of reactions, how we need primers. We talked a little bit about Taq, DNA polymerase, thermus aquatimus. We talked about how that is needed because of the rising temperature in order for that enzyme to stay stable in high heat in order to break the hydrogen bonds. And we talked about how it has to go through multiple iterations, multiple rounds in order to amplify DNA, and how we have to amplify DNA in order to get a good reading on it, right? Something called LODs, limits of detection. We need this thing so we can detect. Just by getting a little swab may not be enough DNA. And we may be looking at specific DNAs. Remember those short tandem repeats? So we talked about short tandem repeats. And it's very important that we go back and look at those notes. So it's very good that this book presentation or ebook starts out with DNA structure to replication, and then now we're going to go to genes and protein. And the reason I really like that is because it follows the theme of the central dogma, and the central dogma being that DNA goes to RNA and RNA then to protein. So DNA to RNA, RNA to protein. That is the central dogma. Make sure you remember that, since it's the pillars of which biology stands on. So it's super important that you have that concept down good. So now in chapter 8, we will be looking at genes to proteins. Okay. And we'll start through that process. And it's an interesting chapter. I'm going to be introducing a few concepts that are going to help bridge the gap of knowledge, because I'm sure it's not all of you read up on genetics and genes and proteins. We'll go through some basics. Again, some stuff that we've covered in chapter 2 and chapter 3. We'll touch up on that, and then we'll get into more details. Okay. So I'm going to start to share screen. Let me first to open up the PowerPoint. My bad. I had to download it. Do we have any questions while we're downloading your PowerPoint?
Nope.
Sir.
Okay. All right. Now I can share screen. All right, I'm going to minimize here. Awesome. Can all of you see the presentation enlarged?
Yes.
Great.
Yes.
All right, so here it is.
Yes.
Thank you so much. I really do appreciate that. Chapter 8, genes to proteins. I'm going to skip this slide. We'll come in. We'll deal with this in a little bit. So let's just talk about the questions, and then let's get into the basics. So as we proceed through this presentation, this chapter, we need to ask ourselves a few questions. At least these are some of the main ones we want to kind of keep in our minds. What determines the shape of a protein? What are the steps of gene expression, and where do they occur in the cell? Now, of course, I don't read it just perfectly like this. I twist it around for my own liking. How can organisms be genetically modified to produce recombinant proteins? What are some pros and cons to genetically modified organisms? We will pass this slide. We'll come back to it. I want to start here before we start getting into this. We can't really go into this until we start really understanding this. So let's get into the protein. This is a recap of what we did in chapter 3. So chapter 3 was our cells, and we bring in proteins and so forth. So again, remember it being one of your macromolecules like carbohydrates, lipids, and proteins, right, as a macromolecule, and nucleic acids being a macromolecule. However, nucleic acids having very, very little energy. So not considered one of the forms of macromolecules used for deriving energy. There were carbohydrates, lipids, and we had some proteins. So again, it is a macromolecule, and it's repetitive. So what are the monomers? What are the subunits? So remember, the word monomer and subunit, same thing. So what are the subunits? What is the monomer? Or as I like to tell you guys, because we all know what Legos are, the little pieces of Legos. We're going to put a bunch of little Legos all bonded together. What is that? Those are going to be amino acids. Amino acids make proteins. All right. Now, again, all enzymes are proteins. All enzymes are proteins, but not all proteins are enzymes. All enzymes are proteins, but not all proteins are enzymes. Enzymes usually ending with the last 3 words, ACE, A-S-E. So what we have here, and we'll look again, we'll dive into it. An amino acid. An amino acid is going to have this common structure right here. You have your carboxyl, hydrogen, and the amino group. And I really like how this book shows that green and then depicts all this green, which is this, and it doesn't change. What changes in amino acids, let's say our 20 amino acids, is the side chain. That is what changes. So for example, CH3, that is what's going to change. All right, CH3. Now, if you come over here, you have your CH3, and then, oh, look, that's an H, not a CH3. Look, it has a new 3-letter code. What does that stand for? Glycine, okay? I'm not gonna ask you for the coding of 3 letters. Don't worry about that. That's for like biochem. But for here, at least you now understand the backbone of it, or what does not change. Once we have these amino acids bonding, they begin to interact because of their side chains, or they begin to withdraw from one another. These side chains, and we won't get into it too much, but they are going to be either polar, non-polar, or there may be even some like neutral characteristics that are all going to dictate its overall 3-dimensional structure and its folding. Once it has this overall structure, then it will have its function, right? Remember, structure is function. Everything in the universe, depending on its structure, it will have a particular function. Same thing with an atom. Why does this element behave like this? Why does that element behave like that? Hey, it's electron configuration, or dictating its overall characteristics, right? So even at the subatomic particle, we see, or subatomic level, we see that structure leads to function. That's what this is. This is, I said a little more than what this says there. Polymer assembly. Here we have what we went into beta pleated sheets. We talked about this stuff here. So we're looking at the overall polymer assembly. So that's our recap of what a protein is. Now, what you're seeing here is that what they're trying to depict is that spiders have a silk, right? Like the web. it's very strong. Like here, they're trying to depict this web. It's very strong protein. And polymer, it's lightweight, it's very super strong, and it can capture insects, and they can't get out. In some cases, some very large spiders in the Amazon, they catch birds. So again, building blocks of proteins are going to be the amino acids, the Legos, the polymers, the subunits. Those are the amino acids. 20 different amino acids. Again, I already talked to you about the core being the carboxyl, the hydrogen, and the amino group. What makes them unique and different from one another is the side group right here. This is all stuff I've already talked about here in terms of 3D conformational shape, the shape, this function, so forth. So this is additional bullet points for you. Now, a very important question. Where do proteins come from? Well, there has to be an instruction manual. You're going to need something to help you figure out like how and sometimes when to make certain things. Instructions that make proteins are encoded by a gene. Okay, these genes. So a sequence of DNA that contains the instructions that make one or more proteins, that is a gene. Okay, and these genes are within the chromosome. So the chromosomes have many genes. And each gene contains instructions to make at least one protein. Okay, so each gene can be expressed or silenced. In other words, it can be turned on or it could be turned off. It can have a green light or it can have a red light. It depends. So let's give an example of what that means. All right. Let's say most of us have heard of lactose intolerance. Anybody here lactose intolerant? Okay.
No, I'm fructose intolerant.
Okay. Right. However, we have an idea more or less what that means, right? Lactose intolerance.
Yes.
Yeah. So some people say it's not able to process, they're not able to process milk well, right?
Yes.
Well, what is it in the milk that's causing people the problems? There's a certain sugar. What's that sugar called? Lactose. If you're lactose intolerant, it's the sugar lactose. What is lactose? Lactose is a disaccharide, two sugars combined together. And what are the two sugars? Glucose and galactose. They're held together, I believe, by beta-1,4 glycosidic linkage. And our body cannot break down lactose. Our body cannot use lactose in any metabolic pathways. For example, anabolism, catabolism, or we want to just call it metabolism. What happens is we need to break apart these two sugars, move them apart. What's the thing that moves these sugars apart? It's an enzyme called lactase. Anyone who does not have this enzyme lactase may be considered lactose intolerant, meaning they cannot tolerate lactose sugar in their diet. Is lactose sugar only found in milk? No, it's found in other dairy products. It could be found in chocolate candies. It could be found in other things like cheeses and just some other dairy products or cottage cheese and so forth. Yogurts. So that is a wonderful example to talk about genes turned on and turned off. Because I became slightly lactose intolerant when I hit 21. What does that mean? That means before I hit 21, I could have all the dairy products in the world. Nothing would happen. My stomach felt perfectly fine. But then something happened at 21 in terms of gene regulation or gene expression. What happened was that the genes that code for me to produce the enzyme lactase started becoming silent. They started to become turned off. Now, I am not severely lactose intolerant. I can drink a glass of milk, I can have cereal, I can have lasagna, I can have cheeses. But if I eat a lot of chocolate, if I eat a good amount of chocolate have higher concentration of lactose sugar. Oh, that can hurt my stomach. And it usually results in a form of like diarrhea. Why? Well, since the body cannot use up these two sugars and it's further down the digestive tract, it is far easier for the body to get rid of it by flushing it out than to bring it back up. Trust me, you don't want something that's that far down to really come back that far up. So it's easier to just create a natural laxative and flush it out. Now, with that said, what about the people who are lactose intolerant and then all of a sudden they're no longer lactose intolerant? Wow. Is it like a miracle? Did something happen? What's going on here? It just means that the body turned on those genes. Okay. So that's a very good example. Now we get into a little bit more interesting questions when we start thinking about, hold on. I think I'm going to turn on this light because I'm getting a lot of reflection on my glasses. Hold on. I think we start getting into some very interesting conversations when we are like, when I say, oh, I went lactose intolerant at 21, no big deal. I'm not gonna die if I don't have dairy products that contain lactose, right? I can get other nutrients elsewhere, right? But let's talk about a child, a baby born lactose intolerant. Now that is a more interesting conversation to have. Why would a child be born lactose intolerant when children, specifically, right, mammals, need their mothers for milk? It makes absolutely no sense whatsoever. So let's rewind 500 years ago. Now, don't lose track of the theme here. This is not me going off into the deep end, in La La Land, on my own little story time. I'm trying to help you understand gene expression, that genes can turn off and on sometimes. Sometimes when they turn off, they cause serious diseases. Now, lactose intolerance is not one of them. But let's talk about this child born 500 years ago, lactose intolerant. How would you feed this child? This baby, not a child, a baby, an infant, a newborn. How would you feed this newborn? Any takes? $500 years ago. Could you go to the store and get formula? 500 years ago? You understand that like 500 years ago, the United States wasn't even founded. There was no United States 500 years ago. So clearly there was no store. So
They were breastfed. So would that have had anything to do if they wouldn't have had to be lactose, right? If they were breastfed, then whatever their mom had, they had too, right?
No, like for example, children of today and in 2020, children that are babies that are born lactose intolerant that can't have, and diarrhea ensues because they can't use it for any metabolic pathways. So what do we do in 2020 to babies that can't have mother's milk? Not because they can't suckle, but because they are lactose intolerant. What do we do?
Well, now they have different formulas.
Yeah. Formulas. Yeah, we have different things that they can handle. Right. So what did people do back then? 500 years ago? Like they didn't even have the invention of plastics. There was no bottles. There was nothing like that. What did they do? They died. A lot of them died. That's just the way it went. Unless you had the resource, the means, and the knowledge to make some sort of like a rice milk. Right? Doesn't happen. So gene expression has serious consequences. Over the time, as technology increased and we had stores and you could go get formula, those children that were lactose intolerant can then grow up and transfer on that genetic information that coded for, or gene or lack thereof, for gene coding and expression of lactose gene. So now we have more lactose intolerant individuals in the human population than 500 years ago, because our technologies can allow it. So genes can turn on and turn off. Last example for helping you understand genes can turn on and off is cystic fibrosis. Cystic fibrosis has, there is a gene called the CFTR gene. It's the cystic fibrosis transmembrane regulatory gene. Individuals that have a mutation, or let's say that gene is not coding correctly what it needs to do, have a form of cystic fibrosis, and that form of cystic fibrosis varies depending on the level of the mutation, which means that individuals do not have the mucosal ciliary escalator. What does that mean? That means that oh no, where did I go? Did I mess up? There we go. Yeah. So what I'm sharing with you here is gene expression. Right here is a healthy individual, and you see this cilia. They're like finger-like projections, almost look like little hairs on the cells of the lungs. They help move along this like watery mucus toward the upper part of the respiratory tract. It's called the mucosal ciliary escalator. And then you can cough and spit and so forth and get this out. When that CFTR gene is affected negatively, the ciliary, or known as mucosal ciliary escalator, it affects this cilia negatively, and there is a larger accumulation of mucus, which then results in the accumulation of bacteria. So the only forms of treatment for an individual that has this gene that's not functioning properly is oxygen, sometimes a compression vest, sometimes physical therapy, in which they revert them, they put them down, head down, kind of an angle, and they kind of pat their backs to help release this mucus. And probably their best option is a lung transplant. That's it. Lots of antibiotics because of a gene that is not right. So it's not turned on right. It's kind of turned off. So I'm just gave you two examples. One with the lactose, in which we've all heard, but we quite don't understand it sometimes. And then now with cystic fibrosis, which we took it more extreme to an actual clinical disease. Okay. Do I have any questions so far? Lactose intolerance questions? All right. So there we are. Chromosome, genes, proteins, right? And we'll dive a little more in depth a little bit. So again, the synthesis of a protein from a gene is called gene expression. Gene expression, what we're kind of talking about. Now we're going to get a little bit into the spider silk. I'm probably assuming, oh, okay, for sure this is an actual spider producing spider silk. Okay, very zoomed in. I'm assuming this is some sort of a scanning electron microscopy. It doesn't say, I wish it would. I wish it would give me some dimensions so I could know what's the actual size of this, an estimation. It doesn't. But I'm telling you that this image is conducted on some sophisticated microscopy. That I know for a fact. So silk starts as a protein-rich liquid made by cells in the silk gland. All right. And then we have them, there's a spinning of it, right? Where they begin to pack tightly in these little spigots coming out. And this is spider silk. Now, what are some of the characteristics of this? Spider silk is very tough. It's flexible, right? It has a lot of applications. Preview this here. Probably the one you're more familiar with is bulletproof vest. Not all bulletproof vests have spider silks involved in them. Some of them use plates, metal plates. Some of them are now using more of a different type of different materials that are not metal. And then in some cases, very lightweight spider silk to help prevent the bullet from passing through the vest and impacting vital organs. So here are some other examples in the applications of spider silk. So now we have, let's get into a little bit more detail. Like, okay, we're talking about some of those applications of the use of spider silk. So are we raising a bunch of spiders somewhere? Are we in a little spider farm and like, I don't know, coming back and picking up the webs? No, we're not doing that. But we're manufacturing them. What say you? Did you say manufacturing? Absolutely. We are manufacturing them. How are we manufacturing this? Oh, let's take a look. All right. So making genetically engineered spider silk begins with isolating the spider DNA. So let's back up before we get into that. So we're going to go to the end product. So here we are first. Here they are making spider silk from yeast cells. And here you can see the purified spun is into this silk fiber. So here they are making that. Okay, let's take a look into the how. Well, we're going to use yeast cells. So we're going to go, I said we had to know what's going on in the end. So let's talk about the how now. Well, we need two things. We're going to be using yeast, and then we're going to be getting the spider. Next question should be, we know we need the spider to get that gene, but why in the world would we use yeast? We understand yeast. Yeast is used in the food industry. We know how to grow it. We know how to wield it. We understand its DNA vectors. We get it. We understand it. So it's a very good subject to be used in recombinant DNA technology or genetic engineering. So what do? We have to isolate the genes that we want and then insert them into the yeast cell. So for example, right here, is the gene of interest from the spider. And here is a gene known as a regulatory gene. Okay, we're going to combine these two. Of course, ladies and gentlemen, this is the simplest terms I can break it down to. We're going to combine them to create a recombinant gene, which then will be inserted into a DNA vector and into the, that's inside this yeast cell. So the recombinant DNA is inserted into a small piece of DNA. This recombinant DNA is added to the yeast nuclei, where it becomes part of the yeast chromosomes, which are now going to produce the regulatory sequence and the spider silk sequence, which is the recombinant gene. Anytime that organisms that have been genetically modified to contain genes from other species are called transgenic. They are called transgenic. Now, some of you may be wondering, what? How? What is going on here? Are you serious? Yeah. How long have we been doing this? It's been a while. We've been doing this for a while. It's in our food industry. Anytime you go and you see something that says GMO, that's called a genetically modified organism. However, I've never really liked the term GMO because it's not very descriptive of what it actually is. It is genetic engineering. Look at this definition again. This is not my definition. This is your book's definition. Organisms that have been genetically modified to contain genes from other species are called transgenic. So that means that we have food that we consume that is genetically engineered, transgenic, because we are a growing population. Now, before I even begin to discuss this part, I want to just state that there are pros and there are cons to this in terms of food, right? We have a large growing population. There's no doubt about it. Just drive from San Antonio all the way to Waco on I-35 in the weekend, and you'll see it was not like that 5-10 years ago. Do it now, and you'll see. So definitely there's an increase of human population. We have to feed. We have to provide food. Can you do it without going GMO? Can you do it without genetic engineering? Yes and no. It depends. Depends on the location. Depends what you're trying to grow. Depends a lot of factors. Now, is there a need to do classical classical breeding? Yes. Is there a need for genetic engineering? I think so. I think we should have both. Specifically, also the classical breeding to increase biodiversity. I think that's a very good thing to have. But there is no doubt that we need food, and we need a lot of it. And not only that, but we export it. So we need things that are going to be drought resistant. We need things that, let's say, the stems are going to be strong so the wind doesn't knock it down. We need things that are going to be resistant to fungi. We need things that are going to be resistant to insect herbivory actions. We need all these things in our food system. And most people don't understand it because most of us are so far removed from agriculture. It used to be that your grandparents or your grandparents, they all grew something at their homes. Most Americans don't grow anything. So we're so far detached from where our food comes from. You talk to little children and they'll tell you that they just think it just grows. Some of them think it just grows on the aisles. It just grows there. It's just there and it's always going to be there. So there are pros and cons to the technology. Well, I haven't quite mentioned what are some of the cons, well, or some of the downfalls. How about allergenicity? You're inserting something from one species into another. For example, let's say corn. Let's say you're inserting the Cry1AB crystal protein from Bacillus thuringiensis, BT bacteria, into corn. Yeah. What if some people have an allergenicity response or allergic response to this foreign protein? Possible, right? So it's just one of those things. It is a complicated topic. It is a heated topic. You know, some people are get very heated about both sides. I personally look at it as there are pros and cons. And the way I look at it in terms of that is that there needs to be really good chronic long-term toxicological risk assessments when we're trying to evaluate food safety in terms of genetic engineering, i.e. GMOs. So that's going on there. And in case that blows your mind about food, and we're looking at, so I think all of us understand that we are in a pandemic. So there is very good evidence indicating that COVID-19 was engineered. So we've been playing with this engineering thing for quite some time, ladies and gentlemen, right? And I think all of us kind of have when I show you this information, we can kind of connect the dots and be like, you know what? Yeah, I get it. There are things out there that we are doing that can positively or negatively affect human population. Again, I talked to you about the regulatory sequence. That's the on and off, the green light, red light, turning genes on and off. The coding sequence determines the identity of the protein. And again, the sequence is going to lead to a structure, which then leads to a function. So to make this transgenic silk, scientists fuse the spider silk, coding the sequence with the regulatory gene. That's what I was saying here. The combination, I already mentioned, this is just additional bullet points, recombinant gene. And in here, we're talking about the vector is the DNA molecule used to deliver the recombinant gene into the host cell, in this case, yeast. All right, let's take a look at gene expression. So gene expression, the overall view of gene expression. So process of converting genetic information into a protein. The process of converting genetic information into a protein. Just one second, gonna look for my phone. Because I can't see the time when we're on our presentations like this. And I wanna make sure I give you the break when it's that time. All right, gene expression. This is a very, very good slide to help portray central dogma. We go from, I said it is, central dogma is, I said from DNA to RNA to protein. So we look at it. DNA, RNA to protein, right? DNA to RNAs to proteins. Central dogma, all right? So here we have DNA. And when we go from DNA to messenger RNA, this is called transcription. So it's going to copy the coding sequence of DNA into a complementary messenger RNA. Messenger RNA. Again, remember, once we start playing around with RNA, we replace that T with a U, right? Uracil. We start that it was a big difference between DNA and RNA. Now, then we have messenger RNA, And messenger RNA then gets out of this nuclear pore into the cytoplasm. Of course, this is taking place in eukaryotic cells. This is not a bacteria setting by any means. So this messenger RNA comes and brings this message to the ribosome, and ribosomes make proteins. This could be a free ribosome. This could be a ribosome with the endoplasmic reticulum, rough endoplasmic reticulum, but it is a ribosome. So what happens is it comes here, and at this point, right here, when you go from the message, now creating the protein, you have translation. Okay, so up here you have transcription, then you have translation here, all right? Now, it's like a code. One of the things I was telling my morning class, I recorded their video, but I'm gonna put yours up because I think you noticed I usually put theirs, but it's just the reason I put their videos up more versus yours is because, one, they're in the morning. So I want to give you additional time to view the lecture. So that's pretty cool. And two, they're just way more like enthusiastic. So there's more students talking and stuff. So I think it's just more entertaining for everybody to watch and hear them. But we're going to put yours up. So we have that situation where there's this code. And I was telling them that when I look at that picture, it reminds me of some history. It reminds me that there was something known as the, during World War II, the Germans had a code, very sophisticated code. And they were using this code to communicate with U-boats, which were like submarine boats, and they would take out cargo ships. The United States was sending supplies into Europe, food, parts to tanks, whatever, arms, medical supplies, and they were knocking them out in the ocean. They would hunt these cargo ships in packs, usually called wolf packs. And there was a code, and it was called the Enigma. And the Enigma was a machine, very sophisticated. So the code would come through into the Enigma, and the Enigma would then print out a little paper, and it had certain codes. The Enigma is like the ribosome, you know? It's like your cell phone. You don't receive, you receive information, right? You receive information, transcription is happening over here at the cell phone tower, then it comes here, and then you have translation. It's translated for you into the protein. You don't get gibberish signal like that we can't interpret because it's translated like transcription. So when you look at things like this, use your imagination. I think that is probably one of the reasons I think I succeeded in school was because I had imagination. I wasn't clearly not the smartest person in my class. Graduate, I didn't even make top 25 in a 100 graduating class. So when you look at this situation here, you got to use your imagination. And there's so much things we can use in terms of like our cell phone technology, what we're looking at right now in our laptops, all this coding, it's happening even on the cellular level. Now, what we'll do is I'm going to stop it here. I think this is slide 19. Yes, slide 19. I'm going to stop it here. I'm going to stop the share and stop the recording. We're going to come back and pick up on 19 when we come back because that one I want to take a little more time to address. So stop recording.